In 2017 alone, 5,295 million metric tons of carbon dioxide were emitted into the atmosphere by the United States. Such emissions have adverse effects on our Earth, as the carbon dioxide traps solar energy within the atmosphere, raising the global temperature and causing many changes, including altered weather patterns, changed growing seasons, and melting of Arctic ice caps. Our oceans play an important role in reducing the amount of carbon dioxide in the atmosphere. Beneath the surface, phytoplankton carry out carbon sequestration, taking in this carbon dioxide and sinking it to the ocean floor. That said, not all of this carbon dioxide reaches the ocean floor, as other microbes, such as bacteria and archaea, intercept the sinking plankton. They degrade this organic carbon and re-emit carbon dioxide back into the atmosphere, thus depleting the effectiveness of carbon sequestration. The current cycles of phytoplankton intake and deposition of carbon dioxide and bacterial re-emission of this carbon dioxide will be interrupted by global warming. Thus, important climate change research is achieved by analyzing these communities and predicting future biogeochemical gradients. The traditional method of analyzing these microbial communities has involved the 16S rRNA gene, a highly conserved gene within species of bacteria and archaea. This gene has 9 hypervariable regions, separated by constant regions. Primers corresponding to one of these constant regions are used to selectively amplify the 16S rRNA gene through PCR, and next-generation sequencing technologies are employed to sequence these genes. Species identification can then be carried out using BLAST. However, while this method is convenient and common, it has many setbacks. PCR has proven to be a problematic process, as primer bias can occur, selectively amplifying some sequences more than others, resulting in skewed population distributions. Amplification also occurs more slowly on lower abundance taxa than on higher abundance taxa. Thus, PCR will inherently provide a misrepresentation of the population distribution, overrepresenting taxa originally present at high abundance levels, while underrepresenting taxa originally present at lower abundance levels. It is hypothesized that metagenomic sequencing, or sequencing all of the genetic information rather than targeting a specific gene, and then utilizing computer algorithms to computationally identify sequences corresponding to the 16S rRNA gene, would provide more accurate population distributions. Here, I examined what, if any, taxonomic differences result from these two approaches. Doing so required 4 main steps. First, sample collection was done by Molly Mikan for her thesis project in 2 Arctic locations, the Bering Strait and the Chukchi Sea. At each location, CTD, conductivity, temperature, and depth, rosettes were lowered into the water. These containers are used to collect water at different depths. A total of 107 liters of water was collected at each location and then pre-filtered using 2 filters to remove larger organisms. Following filtration, 7 layers of the water were further filtered and resulting microbes were sequenced. All the sequences were then grouped together to produce 2 metagenomes, one for each site. Second, Molly Mikan carried out traditional 16S rRNA analysis on the samples, and the data she produced was later used as a comparison for the data derived through metagenomic sequencing and the use of rRNA in this study. Third, rRNA was used to analyze the metagenome. Prior to running rRNA, I acquired access to a computer cluster, or a set of connected computers that work together. This allowed for the processes run by rRNA to be split up and run simultaneously on different computers in order to decrease runtime. rRNA recommended parallelizing about 10 computers, so I requested 10 nodes. Second, rRNA's 2 dependencies, rejoiner and infernal, were added to the user's directory and made available in the current session. Once rRNA's dependencies were loaded, rRNA could then be executed. The output of rRNA was a file that contained the sequences of all the 16S rRNA genes that the program identified and extracted from the metagenome. While there is a second process rRNA could have ran, it is extremely inefficient. Therefore, an alternate approach was devised. In order to match each sequence returned by rRNA to its taxa, a nucleotide blast was used. This compared each unknown sequence to a database of known sequences in order to produce an alignment file. The user must provide the database to compare to, the query or file of unknowns, the name of the output file, the format of the output file, and the number of threads to run the process on. This output file contained one hit for every unknown sequence, indicated by a GI number. A GI number is a number assigned by NCBI and is unique to a sequence record. The file was then opened in Megan, a microbiome analysis tool, where taxonames were assigned based on this number. Megan was then used to interactively visualize the data with phylogenetic trees and bar plots at the class level. Following this analysis, a stamp file and a metadata mapping file were both exported and loaded into STAMP, a program which performs statistical analysis on the datasets. Overall, metagenomic analysis by Riego produced 24% more reads than 16S rRNA analysis. For the Bering Strait, this was about 185,000 reads as compared to 150,000, and 235,000 reads as compared to 190,000 for the Chukchi Sea. While in both datasets a large number of identified bacteria were common between the two methods, unique bacteria were identified for each method as well. Some bacteria were extremely underrepresented in the 16S data as compared to the Riego data. Most notable are the chlorobi group, as in the Bering Strait they composed about 38% of the entire sample from Riego, whereas they only composed 5% of the sample derived from 16S rRNA sequencing. The Chukchi Sea data had a similar outcome, with a 20% abundance of chlorobi group from Riego compared to only about a 2% abundance from the 16S rRNA sequencing. Therefore, it is concluded that the two methods produce different abundance distributions, with 16S rRNA sequencing producing an underrepresentation as compared to Riego, although further study will be carried out to determine which is more accurate. Overall, these results were unexpected, as 16S sequencing identified more diverse taxa, while it was anticipated that the metagenome would have more diversity. Thus, the original hypothesis was both supported and rejected, as its suggestion that if taxonomic distribution was obtained by metagenomic sequencing, then it would be more comparable to the 16S rRNA sequencing data in terms of taxa present was refuted, while its suggestion that taxa would be presented differently in terms of abundance levels was supported. These unexpected results could be because the data used in metagenomic analysis and the data used in 16S rRNA analysis came from two different samples, rather than one combined sample. Bacteria produce different amounts of DNA and RNA based upon how fast they grow and what phase of the growth cycle they are in at the time of collection. As the methods analyzed two different samples, the same class of bacteria could be present in both but at different phases of their growth cycle, causing one method to report a higher abundance level due to more RNA present than in the other. Most likely, though, the discrepancies in the results between the two methods were due to the use of different BLAST platforms. While analyzing the 16S rRNA sequences, BLAST-X was used. BLAST-X takes nucleotide sequences as inputs, splits each one into 6 reading frames, converts them to amino acid sequences, and then compares these sequences against a protein database. While analyzing the metagenome, however, BLAST-N was used, which simply aligns the nucleotide sequences in the input to nucleotide sequences in the nucleotide database. Due to the differences in the methods these two platforms employ, the results each produces are not necessarily comparable, especially as it is believed BLAST-X will produce a greater alignment due to a greater diversity of bacteria. This poses an explanation as to why the Riego results, which were analyzed using BLAST-N, had less diversity than the 16S results, even though the opposite was expected. Due to the unexpected nature of the results and the potential discrepancies due to the use of two different platforms, BLAST-X versus BLAST-N, these results will be treated as preliminary results, and this research will be continued. The next step will be to reanalyze the metagenomic data, this time using BLAST-X to identify the taxa corresponding to the 16S rRNA sequences in the output from Riego.